Good morning, students. Welcome to the morning session of this paper, that is MBT 401. And I'm supposed to take the first unit regarding the animal cell culture techniques. And today I am taking up the introductory class for this particular unit. So, what is animal cell culture? Particularly, why we need it, where can we use it, whether it is ethical or not. All those issues need to be considered when we are talking of the animal cell culture. So, first we take up with the basics or the introductory. This session or this talk will be an introduction to the upcoming lectures. Uh, so we take up this first of all. What is animal cell culture? Now, as you can understand from the name, or that these we are trying to cultivate, or we are trying to culture the cells of animal origin. Now, from where are we getting these cells, or from where are we trying to culture these cells? So the cells are from the animal origin, and they have been isolated or removed from the animal tissue or whole animals. Okay, so they have been isolated from the animal tissue or a whole animal. Just in case, just today we see that there are biopsy samples for cancer patients and all that. Yeah, or you take out blood samples. So you are taking it from living beings. So these cells are mostly living cells. They are derived from the living live tissue from the live animals, and they are being put outside the living animal in the presence of some nutrients and some growth factors and allowed to grow. So basically, when you are trying the cells to grow, uh, to make them grow in presence of some nutrients and growth factors outside the animal outside the animal body, then that particular process is known as your animal cell culture. Now, it always occurs in vitro. When I say the cells are removed from the animal tissue, or the whole body of the animals, that means the process has to be conducted within the uh, within in vitro resources. That is, there are two resources: in vivo or in vitro. In vivo is, that is, you are doing it within the living system. The organism is living, so you are doing the process in the living system. Just like earlier we used to vaccinate people, and then antibodies were grown there, and then the antibodies were retrieved and inserted into the next person for vaccination purpose. So all that was you are doing in vivo techniques. Now the approach has changed. You cannot do it always in vivo. Now the approach is in vitro. So there is a giant leap, you can say, from in vivo to in vitro. This discovery or this invention was just like stepping on the moon of the mankind. Okay, so the mankind tried to reach the moon. Same way, mankind tried to culture or to make the cells live outside the body of the living system. So it always occurs in the in vitro system. Now, these individual cells, they act as independent units. Just like you have bacteria, yeast, or algae, fungi, small or other small microorganisms, they all have independent living. They work as individuals. The whole body system is within a single cell. Similarly, these cells, they also work as individuals or independent units, and they are capable of division and growth. So you are trying to grow these cells in the growth medium, or a nutrient rich medium outside the living system. So that is, you're trying to culture the cells in vitro. Now, earlier when we used to talk of, it was mostly considered as a tissue culture technique. But now, when we talk of tissue culture technique, the word cell culture has become a synonym to it. Tissues were being tried to being cultured, but if you try to culture or maintain the tissue for a longer duration, tissue is a combination of different kind of cells, maybe of same origin but different kind of cells. So all those cells will have a different growth rate, different nutrition requirements, so you cannot maintain that tissue for a longer duration. The cells won't collaborate together to be in assembly, and they won't grow together. So now the approach has shifted from growing tissues to maintaining single individual cells. So synonymously people use it, but rather it is that you are culturing the individual cells. Now, why do we need to culture the cells? Why grow animals cells? Now, first is you are trying to investigate the cell biochemistry and the physiology. How does a cell react to an environmental change or an outward signal? How will it act or function or metabolize the things within the cell? You need to understand the physiology of the living organism. So cell is the basic unit of life, and if you understand the biochemistry and physiology of this basic unit, you will naturally understand the physiology and biochemistry of the living organism that is made up of single of the cells. So if you want to understand how a particular biochemical reaction occurs within the body, how does the muscles work, how the actin filament moves, how the respiratory system works, how the gases exchange in the respiratory system occurs, if you want to understand all those things, then you need to investigate. Now, if you want to investigate these things, if you do it in vivo techniques, that is within the living system, you have to maintain a good pool of animals, or even in the case, maybe if you are trying to investigate the system in human beings, then you have to have human subjects for this to be available for this investigation. So at last you cannot do it always. It is not feasible on a large scale. It will require large efforts, it will require large financial expenditure. So, and the results would be very much similar if you do this process in the cell culture. So if when you try to investigate the cell biochemistry or the physiology, you can do it very easily in the cell cultures. Then next is you need to analyze the effect of different substances on the cells. Suppose if a drug has been given, a medicine has been given to the person, then that medicine will be first metabolized in the liver cells, then it will go to the target organ and then produce an effect. In that case, the response that you will try to understand will be delayed, and it may take hours to analyze the effect of that particular medicine or a toxic substance also. So if you want to reduce that time duration, if you want to make the process easy and practical, then you can use the cell culture. It will definitely reduce the time duration, it will definitely reduce the expenditure and the effort also. Next is to produce artificial tissue. Now, why artificial tissue? You are hearing so much of organ transplant today, you skin grafts and all that. But why people need to do it? Because of some kind of disease condition. People are trying to hold back the aging process. People are trying to heal the disease conditions. So, all for that purpose, you need transplants. You need to grow artificial tissue. Now, this artificial tissue, when you grow in some other organism, suppose for human use you're trying to cultivate or grow this artificial tissue, then you may be doing it this in some cattle animal or some other kind of animal, and then you will sacrifice this and that animal and extract out the tissue and transplant it in the human being. So you are unnecessarily killing or sacrificing an animal for the benefit of humankind. So you can avoid that situation. You can produce this artificial tissue in the in vitro conditions and use it for the transplant operations. Then, next is to produce biological products from large scale cell cultures. Now, if you try to isolate, suppose, insulin for diabetic patients, then you need large amounts of cadavers, that is, the dead bodies, from where you can isolate this insulin that will be compatible with the human insulin. Now, that means you have to put in large efforts, you have to have many cadavers, and you need to extract that insulin from those cadavers. Still, maybe from hundreds of cadavers you may just get a microgram or a little milligram somewhat of insulin. That won't be sufficient to provide a large population dealing with diabetics. In that condition, you need to produce that insulin in large quantities, and also that insulin should be compatible with the human body system. So, in that condition, you can culture these cells that particularly produce insulin in the cell cultures, and you can multiply the number of cells. If you know the growth rate of these cells, then you can definitely multiply the number of cultures or the cell cultures on a large scale, on an economical scale, and produce insulin to suffice for the requirement of a large demanding diabetic population. So this is one example. Similarly, there may be other examples. You have blood clotting factors, you have the other growth hormones or some other hormones. All those you need to produce. There are large requirements by the medical industry. All that can be done. Not this is basically I'm trying about medical industry, but it is not restricted to medical industry. It needs to be done in the pharmacology also, it needs to be done in the veterinary industry also, it needs to be done in the textile industry also. That is, you need for textile industry if you take, or for confectionery industry you take, you need enzymes, different kinds of enzymes. So, those are the biological products that you can take from large scale cell cultures. Not all the cell cultures that we were doing is simply for bacterial cells or microbial cells. You cannot do that. Recombinant products can be used there, but they may not be feasible, or maybe they may generate some kind of antigenic reactions in the body of the human beings, so they cannot be used clinically, they cannot be taken up by the general population. So to keep away those antigenic responses, you need to have biological products that can that will have no problems when we put them into the human system or in another required system. So those enzymes, those proteins, hormones, all those biological products can be produced on a large scale when we use these cell cultures in large scale cell culture units. Then what are the advantages of the cell culture? First and the foremost thing, if you talk as a biotechnologist, then it is the control of the environment. Naturally, you will do the culture in within a closed vessel system. That closed vessel system, you can regulate the temperature, you can regulate the humidity, you can regulate the gaseous components, the exchange of carbon dioxide, oxygen. So you have various controls over there. Then, as I told you in the first thing, that you require cell cultures for analysis and investigation purposes. Now, if you are trying to investigate and analyze the things and the results are not consistent or reproducible, then your experiments will not be good. You cannot definitely or interpret the results if your results are not consistent with your studies. So, if you have a homogeneous population of a single kind of cells, of single type, that is, if you are example you are taking liver cells only, then all are them are liver cells. Then, if you want to see the metabolism of a particular medicine or a drug or a toxic substance, you can easily identify the effect in that particular batch of cells, and that result will be consistent and reproducible. So, whatever experiments you do, whatever analysis or investigations you do, that will be consistent and reproducible. The next is the greater understanding of the toxicity testing, and in addition, it will be a cost effective approach. So, you have to you know the metabolic processes, you know the pathways, you know the physiological aspects of each and every cell. Then if you are providing some kind of toxic substance to that particular cell, it will would be metabolized in a different way, and the pathway may be different. If you label the components of the pathway, then you can easily understand how the toxicity is occurring. So that will give you a more insight into the understanding of the toxicity testing. And in addition, the toxicity testing will also be very cost effective. You don't have to maintain large numbers of animal subjects or human subjects to understand the toxicity effects. You don't have to maintain the other species for larger kind of understanding. So you can simply do it in vitro technique within a simple flask, and you can understand the toxicity of different drugs, components, or toxic substances. Then next is your reduced chances of contaminants in large scale production of biological products. Now, if you are producing some kind of biological product naturally for a human purpose or for feedstock purpose, then you need to have reduced contaminants. Suppose you have vaccine development program, and if you have just isolated the proteins that could generate an antigenic response rather than a whole genomic component, then the vaccine should not contain, even by error, any kind of genomic component in that vaccine. Only the elements or the proteins that could generate the antigenic response should be present there. No other microbial contaminants should be present there. No bacterial components, no fungal components, no spores, no any kind of contaminants should be present in the large scale production of biological products. And then another thing is, when you try to culture the animal cells, That is, you are dealing with cells that generate or replicate or grow or divide at least in a 24-hour cycle. Okay, the bacterial components multiply in a less than half an hour, approximately half an hour cycle. So, if there is any kind of contamination, if there is a bacterial outgrowth or a microbial outgrowth, then the animal cell will definitely be short of the nutrient supply and it will die off. The production process will stop. So if you do it in a culture process, if the environment is controlled, then these kind of contaminants will also be eliminated. Then next is reduction of animal use. As I have told you, this is really an ethical issue that you are sacrificing the animals to generate vaccines, to generate organs for the organ transplant, or to generate tissue for tissue supplements. So that you are putting the animals into captivity and then you are experimenting with them. Suppose, as a human subject, the person is subjected to the lab conditions and you won't be able to stand for the lab conditions for a few hours even. And you are subjecting the animals for uh, just for your investigation purpose, for the study of cancer. There are many animals that are induced with cancers, so carcinogens, so that they develop the cancer, and then you give the anti-cancerous drug to them for the studies. Naturally, if the drug does not work, the animal will die of that induced cancer, the cancer that has been induced by you by using a carcinogen. You are killing that animal ultimately. So, that is not very ethical or a good practice if you try to maintain the common food web or the common ecological cycle or the ecosystem. Okay, so this gives you an opportunity, the cell culture gives you an opportunity to reduce the animal use. Next is with the mechanization aspect or the expertise aspect. Now, cell culture can be done by robotics. You can mechanize the system. You can make the system a continuous process by mechanization, and there could be a continuous flow of nutrients. There could be a continuous flow exchange of gaseous components. With control of the temperature, of the humidity, and all that. So mechanization is very much easily possible in the cell cultures because you are doing the system in vitro rather than in vivo. In vivo is subject to the body conditions of the organism. Then the preservation and characterization of the cells is also possible. You can preserve the cells for a longer duration, characterize the cells as and when possible. So that gives you an opportunity. Suppose if a particular you have grown a cell culture from an organism and you have preserved some of the sample for further uses, and you start running with a little component, a small component with these small samples, then if by chance your sample is destroyed or damaged, you have a preserved sample somewhere which has been characterized and understood, and you can restart with your investigations and analytical processes. Now, the disadvantages of the cell culture. Now, main disadvantages that when you start culturing the cells outside the body, living system, that is in vitro, then you are subjecting them to environmental changes. The cells that were safely put into an milieu of the tissue system or an organ system have been pushed out from that system into an artificial system or a synthetic system. So that continuous culturing brings about changes in the characteristics of the cultured cells. The cells try to adapt themselves according to the changed system, and in that process of adaptation, they keep on evolving. That means after a few generations, after a few passages or a few subculturing steps, the cells that you get will be very much different from the cells that you had originally taken from the living subject. Okay, so there is very much change in the characteristics of the cultured cells. They become the genetic instability, the phenotypic instability, all that comes in when you do the continuous culturing. Then, if you are trying to do a tissue culture, if you're trying to culture a tissue, then a tissue is a combination or a mix of a different kind of cells. So those different kinds of cells will have different growth rates, different nutritional requirements, so that the technique will favor the fast growing cells. That is, as an individual unit, you can carry on with the culture techniques, but if you try to put it in a tissue assembly, then the culture technique would not favor the process. Okay. Next, the cell behavior and adaptability changes makes the study is difficult. Now, as I've told you, that long-term culturing process causes adaptations and evolutionary changes. The cells keep on adapting and evolving. Naturally, their behavior will also change. So now, if you're trying to understand, example, the behavior of an amoeboid cell, how will it respond to the extracellular stresses? How will it create a defense mechanism? Then that behavior will change in response to, because the system was in the uh, living system, in a blood system. Now here it is in artificial system, in a nutrient media. The behavior will naturally change. So you cannot relate the cell behavior with the behavior of that particular cell in the living system. So it, if you want to make such kind of studies, that how will the cell behave in the in vitro system and in vivo system, there will be a large difference in the studies, and you cannot well relate with them. Next is, in response to the adaptations, there would be change in the intracellular enzyme activities. Sometimes some kind of nutrient would be depleted, or the toxic substances would be accumulating, or your product may be being produced. So the cell is continuously trying to adapt with that system, and it will change the response. Then the quantity that you suppose, if you want to grow cells for insulin production, that means you need large quantities of cells to be produced. Large quantities of the growth media needs to be provided in. So the quantity, the expanse of the process is large, and with that expanse, the cost will also will very be high. Then these systems need a skilled handling. Without a skilled handling, without the proper knowledge or the expertise, the cell culture system cannot work. Even if the system is mechanized, is still to monitor the cell cultures, to maintain those cell cultures, some particular level of expertise needs to be present in the handling persons. Only then the process will succeed. Now, when we come to the early history of the cell culture, there is First time it was developed by rocks. The very first method in 1885, it was developed by rocks. And then you have some explant tissue fragments in 1910. Then Ross and Jones worked on the trypsinization and subculture methods in 1916. Then the ATCC was developed, that is American Type Cell Culture organization, that was worked as a repository for the cell cultures, was established in 1925 for characterization of the different cell types that people were isolating. Then new culture devices were developed by Carroll and Lindenberg in 1930s. Then more of then the introduction of antibiotics was done by Kilova, Krishnishka, Lowberry in 1940s. Then first mouse fibroblast cell line was established, that is L cells, in 1943, and so on and so forth. Then in recent, you can say that tissue engineering has been exploited in 2002, and then 3D tissues and organs have been developed by bioprinting techniques, that is beyond your 2010, and so and so forth. New and new things are being developed with the help of the culture techniques, and the system is still growing and evolving and gaining popularity in the present scenario of the biomedical health purposes. Then how will you maintain a cell culture? So the requirements are: first thing, the cells are adherent in nature. They cling together in a tissue environment. So they require an anchor for support. Then the cells require nutrients in the form of hormones, vitamins, so on so forth. Then they should have an aseptic condition. If the I told you the environment needs to be controlled. There should not be any kind of contamination, whether bacterial, viral, or fungal, any kind of. So the aseptic conditions need to be maintained so that the animal cells grow easily and at an optimum growth rate. Now, first products of the animal cell technology, the very first thing that people tried was the development of vaccines. The first of that developed was of the polio vaccine in the eukaryotic cells. Then there were genetically engineered animal cells for the production of biomolecules. Then the hybridoma cells or the fusion cells were produced for the production of monoclonal antibodies. And there are various pharmaceutical, medical, and veterinary products that were also produced utilizing the animal cell culture techniques. Thank you.